Okay, very good afternoon, my dear friends. This is Dr. PADALA Tirupati, Assistant Professor, Department of Botany, Government Degree College for Women, Jagityal. Regarding of this CPGET free online coaching, especially botany. Today's our eminent resource person, Dr. Tungani Rajitha Madam Garu, Assistant Professor, Department of Botany, Government Degree College, Mahadevpur. So, Madam is going to explain one of the important area in plant physiology. So, we would like to invite today's resource person, Dr. T. Rajitha Madam Garu. Please welcome. I have given a chance to share your screen and start your session, madam. Please welcome. No, madam, you are not audible. మీరు అన్మ్యూట్ చేసుకొని మాట్లాడండి. మీరు మ్యూట్ లో ఉన్నారు.
Okay, sir.
Right.
Now, okay.
Yeah, it's audible, madam. Proceed.
Okay. Good afternoon to all of you. Thank you so much, Tirupati sir, for giving up this opportunity to take off this class. I am very exciting to share my thoughts. Thank you so much, sir, once again. And all of you, good afternoon to my dear friends. Today I am going to take the class about on photosynthesis process. Sir, actually this photosynthesis process is under plant physiology. This in CPGET almost various questions, various types of questions are taken from this photosynthesis process. And actually the photosynthesis is a major part in in plant physiology. Actually, the photosynthesis process means the biosynthesis of food material like glucose, fructose in the presence of light is called as a photosynthesis. The biosynthesis of food material like that fructose or glucose. In competitive exam purpose, they are asking about these which type of food materials are preparing in photosynthesis like glucose or fructose. Okay, these are preparing the presence of light. That process is called as a process is called as a photosynthesis. And the next one, this photosynthesis process takes place mainly in green parts of the plant body, like either they are leaves or the epidermal layers of the stem. Okay, and the next one, mainly the green plants are called as autotrophs. Autotrophs means which are prepares their food material themselves by using of light is called as autotrophs. Okay, in competitive exam purpose also they are asking, the word is autotrophs. Autotrophs means which are prepares their food material themselves by using of light is called as autotrophs. Okay, the photosynthesis process we are taken as in equation form. That one is a 6CO2 plus H2O gives rise to C6H12O6 plus 6CO2 and next one 6H2O. Okay, and actually the solar.
Sorry for the interruption.
Sir,
please, is there any PPT for sharing this today's class?
Some slides are there, sir. The chloroplast structure and the next one
if any slides are there, please share in the Zoom.
Okay, I will share it, sir.
Okay, please proceed.
Okay, sir. Today I was taken as just introduction part only. That's why I was not showing slides. Based on topic, I was show my chloroplast structure and chlorophyll structure slides. Okay, actually the solar energy means the solar light is transferred to earth nearly 168 into 10 to the power of 9 joules of energy is transferred to the earth uh, per 1 hectare. 1 hectare means 2.47 acres of area. Okay. How much of energy? 168 into 10 to the power of 9 joules of energy. The solar energy is transfers to 1 hectare of uh, land. Okay. And the next one, nearly 110 into 10 to the power of 9 tons of energy is utilized by the landed plants. And next one, 29 into 10 to the power of 9 tons of energy utilized by the water plants. How much? 110 into 10 to the power of 9 tons of solar energy is utilized by the landed plants. And the next one, 69 into 10 to the power of 9 tons of energy utilized by the water plants. Okay, from this, out of 168 into 10 to the power of 9 joules of energy, only 1% of energy is utilizing by the overall total plants. Okay. And the next one, in 1778, um, Indian House recognized as the oxygen is released by the photosynthesis process. What Indian House is discovered as a oxygen is released by the photosynthesis process in the presence of light. Okay. And the next one, in 1804, Mayer is recognized as a the light energy is converts into the formation of chemical energy. In the process of photosynthesis, the we are already telling about, we already tells that the photosynthesis is a biosynthesis of food material in the presence of light. Okay, that light energy is converts into the formation of chemical energy. That chemical energy is mainly used for the preparation of food material. This process is discovered by the Mayer. And the next one, in 1905, Blackman discovered as a this photosynthesis process consisting of mainly two phases. The first one is the light phase and the second one is the dark phase. In photosynthesis process, we can identify two phases. The first one is the light phase and second one is dark phase. In light phase, by using of this light, the solar energy is converts into chemical energy like ATPs and NADPH2s. Okay. And the next one, in dark phase, this ATPs and the next one NADPH2 energy packages are utilized to convert carbons into the formation of carbohydrates. Okay, and the next one, in 1937, Robert Hill recognized as a oxygen is released from the splitting of water molecules in the process of photosynthesis. In photosynthesis process, we can identify as a oxygen is released. Normally, we can tells that in photosynthesis, oxygen is releasing, carbon dioxide is observing. But this released oxygen is released from the splitting of water molecule. That one is discovered by Robert Hill in 1937. Okay. And the next one, in 1957, Arnon is discovered as a the energy packages. They are either ATP and next one NADPH2. From these two energy packets, ATP is forming by the union of ADP and next one inorganic phosphate gives rise to one ATP. Okay, one ADP and next one inorganic phosphate binds to form say one ATP. One ATP is consisting of 7.3 kilocalories of energy. How much energy? 7.3 kilocalories of energy is stored in one ATP. Okay, and the next one, 7,300 calories of energy. Either we are also taken as a 7.3 kilocalories of energy. In exam point of view, they are asking questions on this ATP also. Okay, and the next one, this photosynthesis process is takes in the cell organelle of chloroplast. The structure of chloroplast Either these chloroplasts either round shaped or oval shaped or semi-rectangular in shape. They are okay. This one is the chloroplast structure. And next one, either it is oval shaped or semicircular in shape or rectangular in shape. And next one, it consisting of this chloroplast consisting of two wall layers. The first one, the outer layer is the outer membrane, and the next one, this one is the outer layer, and this inner thin wall is the innermost layer. And the next one, these both are very smooth and very thin in structures, and they're consisting of a intermembrane space. Intermembrane space means the space in between outer membrane and inner membrane. That one is called as the intermembrane space. And the next one, the inner side of this inner membrane, they consisting of a thylakoid membrane, thylakoids. These thylakoids arranged as a coins. Okay, and the next one, the overall structure of these coins arranged structure is called as a granum. Okay, and the next one, that structure is called as a grana, and in grana, the individual coin-like structure is called as a thylakoid. Okay, this thylakoid consisting of a inner space, that one is called as lumen. And next one, in this on this thylakoid membrane, the chloroplast, all the pigment structures, either they are chlorophylls and carotenoids and phycobilins, all are arranged on thylakoid membrane. And the next one, the inner space, that one is the lumen. This lumen is mainly used for the storage of protons. Okay, and the next one, we are already telling about the dark phase and light phase. The light phase takes place on thylakoid membrane, and the next one, dark phase takes place on stroma. The stroma is present inner side of this inner membrane space. It is a liquid formation, and they consisting of various types of proteins. And the next one, some genetic materials, all these grana structures. And the next one, mainly this process, this chloroplast, and mainly used for the process of photosynthesis. The questions are asked on the structure of granum, and the next one, light phases takes place on which place, and that answer is that light phases takes place on the membrane of thylakoid, and the next one, dark phases takes place in stroma. This stroma is the liquid formation of inner membrane of chloroplast. And the next one, these thylakoids are interconnected with a thread-like structures. They are called as a stroma lamella. Okay, this is the structure of chloroplast.
Madam. Hello. 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 మేడం వినపడుతుందా నేను మాట్లాడేది?
ఇప్పుడు
మేడం
ఓకే సార్. ఓకే సార్, ఓకే సార్.
ఇది మ్యూట్ లో
ఇంతకుముందు
పడిందా మేడం?
మ్యూట్ లో పడింది సార్, అసలు అది
ఓకే
ఓపెన్
మేడం.
కావట్లేదు.
ఓకే ఓకే మేడం, ప్లీజ్ ప్రొసీడ్.
ఓకే ఓకే సార్. సార్ ఒక్క నిమిషం సార్.
చెప్పండి మేడం.
సార్ స్టూడెంట్స్ ఉన్నారా? ఉంటే వాళ్ళు కొంచెం స్క్రీన్ లో వస్తే నేను డైరెక్ట్ క్లాస్ లాగా చెప్పేసుకుంటాను.
ఉన్నారు, స్టూడెంట్స్ ఉన్నారు.
సార్ నేను
మీరు మీరు డైరెక్ట్ కంటిన్యూషన్ కానివ్వండి, వాళ్ళను మాట్లాడనివ్వకుండా నేను ఏం చేశానంటే వాళ్ళు
ఓకే
అన్మ్యూట్
ఓకే సార్.
చేసుకోనివ్వకుండా చేశాను, ఎందుకంటే డిస్టర్బెన్స్ ఉండకుండా.
ఉమ్,
వాళ్ళు
ఓకే
మీ
ఓకే
క్లాస్
సార్.
అయిన తర్వాత వాళ్ళకి ఓకే మేడం, ప్రొసీడ్.
ఓకే సార్, థ్యాంక్ యూ. Okay, this one is the chloroplast structure. I think ఇంతకుముందు నీవు నా వాయిస్ వినబడక మీది వినలేకపోయారేమో అనుకుంటున్నాను. అందుకనేసి నేను మళ్ళీ ఒకసారి ఎక్స్ప్లెయిన్ చేయాలనుకుంటున్నా అయితే. ఓకే, in chloroplast structure we can identify either it is round structure or semicircular in shape or semi-rounded. And next one, in chloroplast we can identify as it consisting of uh, it consists of two membranes. The first one is the outer membrane. This one is very thick and next very smooth in their structure. This one is the outer membrane, and the next one is inside of this outer membrane. They consisting of a inner membrane. It is very thin and very smooth and very transparent compared to the outer membrane. Okay, and the next one, in between these two, outer membrane and inner membrane, they consisting of a space. That one is called as a intermembrane space. This intermembrane space, this one is the intermembrane space. In this intermembrane space, the protons are stored at this place. Okay, and the next one is the inner side of this. The inner side of this inner membrane space. This one is the inner membrane. Okay, this the inner side of this inner membrane, they consisting of a liquid formation around all the inside of this inner membrane. This liquid formation is called as a stroma. Okay, this stroma consisting of various types of proteins, and the next one genetic material, and next one, this stroma also consisting of which enzymes are useful for the phase. Okay, and the next one, in this stroma, we also identify as a grana. This granum individual is called as, this one is a grana, and this individually called as a granum, and the next one overall combines two forms as called as a granum. Okay, in individual grana, we can identify a coins-like structures. Coins-like structures, they are first one, these are these coin-like structures. They are arranged as one above the other, and the next one, these individual structures are called as a thylakoid. Okay, and these thylakoids are very rounded and flattened structures, and also they consisting of a innermost inner side of these, they consisting of a gap, and that one is called as a lumen. Okay, And the next one, these thylakoids. Upon these thylakoids, they consisting of a various types of electron carriers and as well as a pigments. We already tell about the pigments. That pigments are arranged on these wall layers of the thylakoids. Okay. And the next one, in photosynthesis process, we can identify two types of phases. The first one is the light phase and second one is the dark phase. This light phase takes place on thylakoid membrane and the next one, dark phase, takes place in stroma. Okay. In photosynthesis process, how many phases? Two phases. They are also asking about on these two phases, the light phase and the next one is the dark phase. And the next light phase takes place on thylakoid membrane. Okay. And the next one, dark phase, takes place in stroma. Okay. This one is the structure of chloroplast. Okay. Okay. And next, in photosynthesis process, we can identify three types of pigments. That pigments are arranged on thylakoid membrane. Especially these pigments are, please take down this. First one is the chlorophyll, carotenoids, and phycobilins. How many types of pigments are there? Three types of pigments are there. The first one is the chlorophylls, carotenoids, and phycobilins. These chlorophylls are mainly, there are 9 types. How many types? 9 types of chlorophylls are there. The first 5 are chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D, and chlorophyll E. How many types of chlorophylls are there? 5 types of chlorophylls are there. The first one is the chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D, and chlorophyll E. And the next 2 are bacteriochlorophyll A, bacteriochlorophyll B. Okay, and next remaining 2 are chlorobium chlorophyll 650 and chlorobium chlorophyll 660. Okay, basically, how many types of chlorophylls are there? 9 types of chlorophylls are there. Mainly, the questions also asking on these 9 types. The first one is the chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D, and chlorophyll E. And next remaining 2 are bacteriochlorophyll A, and next one bacteriochlorophyll B, and remaining 2 are chlorobium chlorophyll 650 and chlorobium chlorophyll 660. All these 9 are combined to called as a chlorophylls. Okay, and next one, among these all 9 chlorophylls, only A is the main chlorophyll. Okay, and this chlorophyll A is present all the reaction centers. Okay, remaining pigments absorb solar energy and they transfers their energy to the reaction center. Means they transfers their absorbed energy to the reaction center. That one is the chlorophyll A. And the next one, this chlorophyll A is present all types of plant bodies and all types of which are doing photosynthesis in the all that organisms consisting of chlorophyll A. Exceptionally, bacteria did not show chlorophyll A. That consisting of bacteriochlorophyll A. But this not consisting of a normal chlorophyll A. Okay, means chlorophyll A is present all the chlorophyll A is present all the photosynthesis showing organisms exceptionally bacteria. Especially bacteria consisting of only bacteria chlorophyll A. Okay, and the next one, carotenoids. These chlorophylls are either green color or bluish in color. And the next one, these carotenoids. Carotenoids are reddish or orangish in color. Carotenoids are reddish or orangish in color. And the next one, they are present in, mostly they are present in vegetables. And the next one, phycobilins. Phycobilins are mostly present in blue-green algae, in which they are present in blue-green algae and red algae. We are identify phycobilins. Please note down this point. Phycobilins are present in blue-green algae and red algae. Okay, almostly chlorophylls are present all the organisms. Exceptionally, chlorophyll A is not present in bacteria. This one point. And the next one, carotenoids and carotenoids are mainly they are reddish or orangish in color. Mostly they are present in vegetables and vegetables. And the next one, phycobilins. Phycobilins are present in blue-green algae. And next one, rhodophyceae members. Okay. And the next one, this chlorophyll A and chlorophyll B, almost all organisms consisting. And next one, the ratio of this chlorophyll A and chlorophyll B is the 3:1 ratio. Okay. This ratio is also asking in entrance examination point of view. Okay. How much ratio is showing chlorophyll A and chlorophyll B is a 3:1 ratio. Okay. And the next one, chlorophyll C. Chlorophyll C is mostly present in Phaeophyceae members. In which members? Phaeophyceae members and diatoms. Okay. Chlorophyll C is present in diatoms, and the next one is the Phaeophyceae members. Okay. And the next one, chlorophyll D. Chlorophyll D is present in rhodophyceae members. And the next one, chlorophyll E is present in xanthophyceae members. All these points are asking in examination point of view, entrance examinations. Okay. Chlorophyll A and B is present in the ratio of 3:1. And the next one, chlorophyll C is present in Phaeophyceae members and rhodophyceae members. Chlorophyll D is present in rhodophyceae members, and chlorophyll E is present in xanthophyceae members. Okay. And the next point, the chlorophylls are in the shape of the bat shape. Okay, badminton bat, in which shape they consisting of a round circle at the beginning, and they consisting of a tail-like structure. Okay, this like structure we are identifying in chlorophylls. Mainly, I will show you the structure of this chlorophylls. They consisting of a two parts, head part and tail part. Okay, this one is the structure of chlorophyll. Okay. Mainly, the structure of chlorophyll consisting of the head part, and the next one is the tail part. Badminton, bat shaped. Okay, the head, and the next one is the tail shaped. Okay, and the next one, this head part is called as a porphyrin ring. Okay, what we are calling porphyrin ring. And the next one, this tail is called as a phytol tail. Okay, in entrance examination point of view, also asking about porphyrin, and the next one, phytol. Okay, the head part is called as a porphyrin, and the next one, the tail part is called as a phytol tail. Okay, and the next in head part, we can identify a central. And that one is the magnesium, and this magnesium is surrounded with 4 nitrogens, and each nitrogen surrounds with a 4 carbons. Okay, and the next one, okay, this structure is the structure of chlorophyll consisting of porphyrin head, and next one, phytol tail. In porphyrin head, we can identify a centrally arranged magnesium. Magnesium is surrounded with 4 nitrogens, and each nitrogen binds with the 4 carbons. And the next one, the tail-like structure is called as a phytol tail. Okay, these chlorophylls are arranged. Already we are know these chlorophylls are arranged on the membrane of thylakoids. This one is the thylakoid and individual thylakoid membrane. This one is the individual thylakoid membrane. On this thylakoid membrane, we can identify arrangement of these chlorophylls. Okay, this individual chlorophyll structure is the this one is the structure. It is badminton shaped and it consisting of head part and tail part. Okay, and the next one, the formulas of this chlorophylls also asking in entrance examination point of view. Please note down that formulas is most important. Please take down this. Okay, the first one. This one is the chlorophyll A formula C55H72O5N4Mg. C55H72O5N4Mg. And chlorophyll B is the C55H70O6N4Mg. And next one, chlorophyll C, C35H7, sorry, H32O5N4Mg. And next one, chlorophyll D is the C54H70O6N4Mg. Mostly in examination point of view, they are asking about only chlorophyll A and the next one, chlorophyll B. Okay, mostly they are asking about chlorophyll A structure and next one, chlorophyll B structure. The chlorophyll A formula is the C55H72O5N4Mg. And next one, chlorophyll B is the C55H70O6N4Mg. Okay, please screenshot this slide. Okay, these are the structures and types of pigments. All these pigments are present in chloroplast. And the next one, in photosynthesis process, we can identify mainly, we are telling like that, biosynthesis of food material. Okay, food material means either the glucose or fructose. They are preparing by the presence of light. Okay, actually మేము ఏమనుకుంటున్నామంటే ఫోటోసింథసిస్ ప్రాసెస్ అనేటువంటిది ఒకసారి జరిగేసి దాంట్లో ఫుడ్ మెటీరియల్ అనేటువంటిది ప్రిపేర్ అవుతుంది అని అనుకుంటున్నాము. కానీ ఏమవుతుందంటే ఫోటోసింథసిస్ లో వీ కెన్ ఐడెంటిఫై 2 phases. They are light phase and the next one is the dark phase. The first one, light phase, takes place on the thylakoid membranes of chloroplast, and dark phase takes place in stroma of chloroplast. Stroma and thylakoids both are present inside of this inner membrane. Okay, and the next one, this light phase. In light phase, we can identify as a the solar energy is directly converted into the formation of chemical energy. Okay, in which phase the light energy is converts into chemical energy? In light phase. Okay, and the next one, in dark phase. In dark phase means it did not accept something. It did not requires any light in the preparation of food material. Especially they uses the energy packets like ATP and next one NADPH2. By using of that energy packets only, the carbons is converts into the formation of carbohydrates in dark phase. Okay, that's why this dark phase and light phase and dark phase both are identified by the Blackman. ఇవి మీరు చాలా గుర్తుపెట్టుకోవాల్సినవి ఏంటంటే ఇండియన్ హౌస్ ఏం చేశారు, అండ్ ద నెక్స్ట్ వన్ మేయర్ ఏం చేశారు, బ్లాక్ మెయిన్ ఏం చేశారు, రాబర్ట్ కిల్ ఏం చేశారు ఇన్ స్టార్టింగ్. I was already tell in the initial starting of this class. And the next one, I, I will show you one diagram of this light phase and next one dark phase. Okay. This actually this total structure is the process of photosynthesis. In photosynthesis process, the solar energy is converts into the formation of the solar energy is converts into the formation of chemical energy. There are ATP or NADPH2 in the process of light. Okay, the solar energy is converts into the formation of ATP and next one NADPH2 in the process of light. This one is the first phase of photosynthesis, and the next one, second phase, is the dark phase. In dark phase, the formation of energy packets in light phase. There are ATP and next one NADPH2. These both are utilized and converts the carbons. The plants absorbs carbon dioxide. This carbon is converts into the formation of carbohydrates in the process of dark reaction. This dark reaction is the second phase of photosynthesis. Okay, how many phases are there? The first one is the light phase and second one is the dark phase. In light phase, the chemical solar energy is converts into the formation of ATP and next one NADPH2. They are nothing but the chemical energies. Okay, especially the solar energy is utilized in light reaction only. Okay, this solar energy is converts ATP and next one NADPH2. The end product of this light reaction is the ATP and next one NADPH2. And they the formed these ATP and NADPH2 both are enters into the stroma of inner membrane. Okay, and the next one, all these reactions, these light phase reactions, all are takes place on the thylakoid membrane, and remaining reactions are takes place in stroma. These ATP and NADPH2 both are also enters into the stroma of chloroplast. And the next one, by using of this ATP and NADPH2, these absorbed carbons are converts into the formation of sugars or carbohydrates. Okay, and the next one, in dark phase, in this process, the light is not required. Only light is required in light phase. Okay, the light is not requiring in this second phase. That's why this one is called as a dark reaction. Okay, and the next one, in this process, if in this total process, we are identified as a oxygen is releasing from the photosynthesis. Okay, the released oxygen is splitted by the water molecule in the process. Okay, this one is the light phase or light reaction. The solar energy absorbing by the chloroplast. In chloroplast, we can identify the chlorophyll pigments. They absorb this solar energy. And the next one, in this solar energy, in this light phase, the water molecule is splitting. Okay, splitting, and next one, two protons and electrons released. These released protons and electrons are utilized by the ADP and the next one NADP, and they converts into the formation of ATP and next one NADPH2. Okay, and the next one, the released oxygen is this one. ఓకే, యాక్చువల్లీ ఇన్ ఫోటోసింథసిస్ ప్రాసెస్ వీఆర్ ఐడెంటిఫైడ్ అంటే మనం ఏమనుకుంటున్నాం, ఫోటోసింథసిస్ ప్రాసెస్ లో ఆక్సిజన్ రిలీజ్ అవుతుంది అని అనుకుంటున్నాం. అంటే మనం ఏమనుకుంటామంటే CO2 కార్బన్ డైఆక్సైడ్ అనేటువంటిది కన్వర్ట్ అయిపోయి ఆక్సిజన్ రిలీజ్ అవుతుందేమో అనేసి నాకు చిన్నప్పటి నుండి ఒక ఊహ ఉంది. కానీ అక్కడ ఆక్సిజన్ కార్బన్ డైఆక్సైడ్ తీసుకునేటువంటి కార్బన్ డైఆక్సైడ్ లోని కార్బన్ వచ్చేసేమో కార్బోహైడ్రేట్స్ ఫార్మేషన్ లో యూజ్ అవుతూ ఉంటుంది. మరి రిలీజ్ అయినటువంటి ఆక్సిజన్ ఎక్కడ నుండి వస్తుంది అంటే లైట్ రియాక్షన్ లో స్ప్లిట్ అయినటువంటి వాటర్ మాలిక్యూల్ నుంచి రిలీజ్ అయినటువంటి ఆక్సిజన్ అన్నమాట. ఓకే, ఈ ఆక్సిజన్ మాలిక్యూల్ గురించి కూడా మనకి ఎగ్జామినేషన్ పాయింట్ ఆఫ్ వ్యూ లో అడుగుతూ ఉంటారు. ఓకే, అంటే ఫోటోసింథసిస్ ప్రాసెస్ లో రిలీజ్ అయినటువంటి ఆక్సిజన్ దేని నుండి రిలీజ్ అయిపోతుంది, దేని నుండి వస్తుంది అని అంటే మనం ఏంటి ద స్ప్లిటింగ్ ఆఫ్ వాటర్ మాలిక్యూల్. ఓకే, వాటర్ మాలిక్యూల్ స్ప్లిట్ అవ్వడం వల్ల వచ్చినటువంటి ఆక్సిజన్ మనము ఫోటోసింథసిస్ లో రిలీజ్ అవ్వడాన్ని మనం అబ్జర్వ్ చేస్తూ ఉంటాం. ఓకే, దీంట్లో నుంచి వాటర్ మాలిక్యూల్ స్ప్లిట్టింగ్ లోనే ఏమేమి రిలీజ్ అవుతున్నాయి, ప్రోటాన్స్ రిలీజ్ అవుతున్నాయి, ఎలక్ట్రాన్స్ రిలీజ్ అవుతున్నాయి. ఈ ప్రోటాన్స్ ఎలక్ట్రాన్స్ రెండు కూడా దేనికి యూజ్ అవుతుంటున్నాయి, ఎన్ఏడిపి ఎన్ఏడిపి కన్వర్ట్స్ ఇంటూ ఫార్మేషన్ ఆఫ్ NADPH2 అండ్ ద నెక్స్ట్ వన్ వచ్చేసి ఏంటి ADP ప్లస్ 1 ఫాస్ఫేట్ బైండ్ కావడానికి కూడా అవి ప్రోటాన్స్ అండ్ ఎలక్ట్రాన్స్ యూటిలైజ్ అయిపోయి ఈ ATP అండ్ నెక్స్ట్ వన్ NADPH2 అనేటువంటి ఏర్పడుతుంది. ఇదంతా ప్రాసెస్ కూడా మనం ఎక్కడ అబ్జర్వ్ చేస్తున్నామో లైట్ ఫేజ్ లో ఐడెంటిఫై చేస్తూ ఉంటున్నాం. లైట్ ఫేజ్ లో ఎండ్ ప్రోడక్ట్ వచ్చేసి ATP NADPH2. ఓకే, and the next one the end product of this light phase ATP and NADPH2 both are utilized in the process of dark phase. ఈ డార్క్ ఫేజ్ లో ఏమవుతూ ఉంటుందంటే ఈ అబ్జర్వ్ చేసినటువంటి కార్బన్ డైఆక్సైడ్ దేని ద్వారా అబ్జర్వ్ చేయబడుతూ ఉంటుంది, స్టొమాటా లీఫ్ లో ఉండేటువంటి స్టొమాటాలో నుంచి అబ్జర్వ్ చేయబడినటువంటి కార్బన్ డైఆక్సైడ్ డార్క్ ఫేజ్ లో RUBP అబ్జర్వ్ చేసేసి అది కాస్త దీనిగా డార్క్ ఫేజ్ లో అబ్జర్వ్ చేసిన తర్వాత అది షుగర్స్ గా కన్వర్ట్ అవుతుంటుంది. అంటే ఏం చేస్తుంటుంది, RUBP అబ్జర్వ్ చేసుకుని షుగర్స్ నథింగ్ బట్ గ్లూకోజ్ గా కన్వర్ట్ అయిపోయి మళ్ళీ తిరిగి రీఫార్మేషన్ ఆఫ్ RUBP ఫార్మేషన్ లో జరుగుతూ ఉంటుంది ఇక్కడ డార్క్ రియాక్షన్ లో. నెక్స్ట్ క్లాస్ లో చెప్పినప్పుడు నేను టోటల్గా లైట్ ఫేజ్ గురించి అండ్ డార్క్ ఫేజ్ లో ఉండేటువంటి ప్రాసెసెస్ గురించి టోటల్ ఎక్స్ప్లెనేషన్ ఇచ్చేస్తాను. ఓకే, యాక్చువల్లీ ఈ రోజు చెప్పాల్సింది ఏంటంటే మెయిన్ గా ఫోటోసింథసిస్ ప్రాసెస్ లో చూసినట్లయితే లైట్ ఫేజ్ ఉంటుంది, డార్క్ ఫేజ్ ఉంటుంది. లైట్ ఫేజ్ లో వచ్చేసేమో లైట్ ని అబ్జర్వ్ చేసుకొని లైట్ ఎనర్జీ కన్వర్ట్స్ ఇంటూ ఫార్మేషన్ ఆఫ్ కెమికల్ ఎనర్జీ అని ఆ కెమికల్ ఎనర్జీ వచ్చేసి ATP అండ్ NADPH2 అని ఈ లైట్ ఫేజ్ అంతా కూడా థైలకాయిడ్ మెంబ్రెయిన్ పైన జరుగుతుంది. ఓకే, అండ్ ద నెక్స్ట్ వన్ డార్క్ ఫేజ్ చూసినట్లయితే లైట్ ఫేజ్ లో రిలీజ్ అయినటువంటి ATP NADPH2 యూటిలైజ్ చేసుకొని కార్బోహైడ్రేట్స్ కన్వర్ట్ అవుతూ ఉంటున్నాయి దీంట్లో డార్క్ రియాక్షన్ లో. ఓకే, లైట్ ఫేజ్ లో లైట్ అవసరం అవుతుంది కానీ డార్క్ ఫేజ్ లో లైట్ ఇలాంటి ఇన్ఫ్లుయెన్స్ చూపించదు. లైట్ ఉన్నప్పటికీ లేనప్పటికీ కూడా డార్క్ రియాక్షన్ అనేటువంటిది సాగుతూనే ఉంటుంది. లైట్ ఫేజ్ అనేటువంటి ఓన్లీ లైట్ టైమ్ లోనే జరుగుతూ ఉంటుంది. అండ్ నెక్స్ట్ వన్ చూసినట్లయితే డార్క్ రియాక్షన్ అనేటువంటి డార్క్ రియాక్షన్ అనేటువంటిది ఎనీ టైం ఐదర్ లైటింగ్ ఫేజ్ లైట్ టైం ఆర్ నైట్ టైం ఎనీ టైం మనం డార్క్ రియాక్షన్ ప్రాసెస్ ని ఐడెంటిఫై చేయొచ్చు. ఓకే, ఇన్ ద నెక్స్ట్ దీంట్లో మనం ఐడెంటిఫై చేసినట్లయితే లైట్ రియాక్షన్ డార్క్ రియాక్షన్ రెండిట్లో చేసినట్లయితే అంటే 12 వాటర్ మాలిక్యూల్ ఆక్సిడైజ్డ్ అవుతూ ఉంటున్నాయి. అండ్ ద నెక్స్ట్ వన్ చూస్తే మనకి 12 ATPs and next one 12 NADPH2 are releasing in light reaction. Actually, how many water molecules are splitting? 12 water molecules are splitting, and the next one by splitting of these water molecules, the end product they are forming how many ATPs? 12 ATPs and 12 NADPH2. Okay, and the next one, this mechanism of photosynthesis also called as the oxidation reduction. Okay, which process this also in examination point of view లో చూసినట్లయితే ఈ వర్డ్ కూడా మనకి చాలా ఇంపార్టెంట్. దీన్ని చాలా కాన్సన్ట్రేషన్ అబ్జర్వ్ చేయండి. స్పెషల్లీ ఇది నేను మెన్షన్ చేయడానికి కూడా రీజన్ ఇదే oxidation reduction. Oxidation means loss of electrons and reduction means gain of electrons. Confusion లో ఉంటూ ఉంటారు students. Oxidation means loss of electrons and reduction means gain of electrons. Okay, ఈ పాయింట్ ఆఫ్ వ్యూ లో మనం క్వశ్చన్స్ అనేటువంటివి అడుగుతూ ఉంటారు కన్ఫ్యూజ్ చేయడానికి. And the next one మీరు గుర్తుపెట్టుకోవాల్సింది 12 automaticals, 12 ATPs, and 12 NADPH2s. And next one light phase and dark phase. Okay. In next class లో వచ్చేసి remaining topics. నెక్స్ట్ క్లాస్ లో ఈ లైట్ ఫేజ్ అండ్ నెక్స్ట్ వన్ డార్క్ ఫేజ్ టోటలీగా ఎక్స్ప్లెయిన్ చేసేస్తాం. ఓకే, థ్యాంక్ యూ సో మచ్ స్టూడెంట్. హలో సార్. Hello, sir. Hello.
Madam, am I audible?
Sir, your voice
Madam.
is not clear. Sir. Not clear, sir.
Now I'm audible.
Hello, sir.
Hello.
Sir.
Audible, madam?
Yes, sir. No, okay.
So, మనం ఈ రోజు వైండ్ అప్ చేద్దామా మేడం సెషన్?
చేసేద్దాం సార్. రేపు అంటే నెక్స్ట్ టైం చెప్పినప్పుడు లైట్ ఫేజ్ డార్క్ ఫేజ్ టోటలీగా చెప్పేసిస్తాను. ఇంకా జస్ట్ ఇంట్రడక్షన్ పార్ట్. ఓకే సార్.
రైట్.
థ్యాంక్ యూ సార్.
థ్యాంక్ యూ వెరీ మచ్ మేడం. Dr. Rajitha madam garu has clearly explained about the main important feature of the photosynthesis in the plant physiology, and most important points madam has covered today class because so many questions has covered in the CPGET examination. In previous papers we are observed minimum 2 to 3 questions, minimum. Maximum 5 to 6 questions has come from today's class only. So that's why today's class is most important. So thank you very much, madam. So remaining class we'll discuss tomorrow. Thank you very much. I will going to wind up the session. Right, madam. Thank you.
Thank you.